Hello everyone and welcome to Module 8. This week we are focusing on a topic that is very true to my heart, as I have spent probably the better part of my entire medical assisting career working in internal medicine. So this week, this is what we are focusing on. And with that, we are talking about two chapters, Chapter 45 and Chapter 48. Now, while these two chapters are what we are focusing on this week, they are not specifically in direct conversation and direct discussion with the topic of internal medicine. So I want to just take just a minute and kind of talk about what exactly internal medicine is, so that way you have a good understanding. The chapters this week are going to be talking about things like microbiology and hematology, which play into the role of internal medicine, but not specifically discussing that. So internal medicine is medicine that provides and providers use to prevent, diagnose, and treat diseases that affect adults. Now, typically in internal medicine, they only see patients who are 18 and older, where unlike family practice, they will see patients from birth to the elderly. So they only see from what would be considered an adult, 18, to the elderly and beyond. Now, because these providers focus on the adult patient, they also must be knowledgeable in the areas of microbiology and hematology. This is why we're talking about those two specific chapters this week, okay? So let's go ahead and get in. We're going to start with Chapter 45, which is on the study of microbiology. Now, microbiology is the study of living organisms that are too small to be seen with the naked eye. So in order to view these microorganisms, we need to use a microscope, kind of where that concept was developed for microbiology. And it was developed by a man called Louis Pasteur, and he's credited as being the father of microbiology, kind of like Hippocrates is the father of medicine. This study has greatly advanced the field of medicine and currently allows individuals to live longer, healthier, and happier lives. And knowing that it is important to understand, we need to understand that we are surrounded by microorganisms. But not all of the microorganisms around us are bad microorganisms. We have some good ones. For example, our bodies make what we call normal flora. And this is actually a beneficial bacteria, as it really helps to use our own body's bacteria that it makes to resist pathogens. So in this discussion and the topic of microbiology, it's important that you as the future medical assistant really understand what your role would be in the topic of microbiology. So as a medical assistant, you frequently will come in contact with blood and bodily fluids. This is why we teach you about standard precautions and universal precautions and the bloodborne pathogen standards. All of those things that you learned in your fundamental course really kind of come back to play when we talk about microbiology. You could be responsible for collecting or maybe even preparing specimens for testing, or maybe you're simply just there to help with patient care during a microbiology procedure. All of this is really important, and you need to remember that it's your responsibility to really stop the spread of infections. Again, using your standard precautions, making sure you're wearing your proper protective equipment, making sure that you are washing your hands between patients and you're cleaning the rooms between patients, and really trying to stop that spread of infection. So now, as we go into microbiology a little bit more, we really need to discuss how bacteria, or how maybe microorganisms, really are classified. In order to work within the field, you will need to really understand how microorganisms are classified. But you as the medical assistant need to know that you are not responsible for identifying and naming specific microorganisms. So when we start talking about how microorganisms are named, they typically have two names. They have a genus name and a species name. Your genus name will always be the one that you see as a capitalized name, where your species is always going to be lowercase. It is very similar to that of, for example, like a brand and a generic name of medications. But again, you are really not responsible nor expected to learn all the genus names for microorganisms. It's simply that you need to understand the terminology that is used and that is necessary to maybe receive lab reports and read them, but also being able to get those results over the phone and having them be able to be delivered to the providers. Now, not just naming them is how microorganisms are classified. They're also classified by the retention of dyes. This means they are characterized by their reactions to certain stains. And typically, a stain is a dye that is used in coloring microorganisms to make them visible under the microscope. Typically, we use a gram stain, and what we do is we actually end up staining the bacteria. If a microorganism turns violet in color, it's considered gram stain positive. If it remains pink in color, it is considered gram stain negative. And lastly, organisms are also classified by whether or not they can survive in oxygen-rich environments or whether they die in the presence of oxygen. If they live and survive in oxygen-rich environments, they are what we call aerobic. Likewise, if they die in the presence of oxygen, they are called anaerobic. So now let's get our discussion going on the different types of microorganisms that are out there in the world. Microorganisms are divided into groups based on the sharing of special characteristics. They could be things such as bacteria, which are the most numerous of all microorganisms and are capable of rapid reproduction. Typically, bacteria are unicellular, so they're one-celled, and they thrive in warm, dark, moist environments like the human body that we all have. Now, bacteria are identified by their shape and appearance, which can be cocci, which is round or spherical, bacilli, which are more shaped like rods, and spirilla, which are spiral-shaped. Another microorganism that we do have is a virus, and this is the smallest of all microorganisms, and it can only be seen with the use of a microscope. Viruses can only multiply within a host or living cell, and they are difficult to kill with medication. That's why you typically do not receive an antibiotic when you have a viral infection. However, viruses can be destroyed with heat, for example, in the use of an autoclave, but they cannot be destroyed by the use of chemical disinfectants. More viruses than any other category are considered microbial agents, and they like to feed on antibiotics, so that's why we typically don't give those, again, during those viral infections. Protozoa are another type of microorganism. They are one-celled organisms. They can be both parasitic and non-parasitic. And they typically move with cilia, or false little feet, or little hair-like productions. And they're typically about 2 to 200 millimeters in size, so very, very tiny. Fungi is our next type of microorganism. These are parasitic, and some can be considered non-parasitic, but typically those are things like plants and mold. Now, fungi depend on the life forms of others for their nutrition, and they reproduce by what we call a budding system. Typically, you will see yeast as a fungus, and again, funguses like to feed on antibiotics. And the last one that we have is the parasites. Now, parasites receive their nutrients from another organism. They can be single-celled or multi-celled, and two great examples of parasites can be things such as worms or insects. So the last topic I want to talk about for Chapter 45 in microbiology is how we actually diagnose these different types of microorganisms. So first, the patient would be examined and information would be gathered. This is again where you as the medical assistant come in. You're going to gather their vital signs, their chief complaint, their history, all of those things that we would do in any other normal visit. But then a specimen would need to be collected and prepared for transportation. So it's important to discuss that when you're transporting microbiology specimens, you need to make sure that you follow the correct protocol. Do not take any shortcuts because it could ruin the sample or the collection process. And there are several types of collection devices that we use, such as sterile swabs, which the shaft and tips of the swabs can vary in size, and different types of materials could be used on those. A culturette is composed of a disposable, clear plastic tube with a sterile cotton applicator, and it's sealed in a plastic vial and has some sort of medium to it. We typically collect things from the throat, nose, and eyes, or the genitalia areas with culturettes. And a smear is when we place a thin layer of the microorganism on a glass slide for identification purposes. This is where we would use that staining process again. Alright, so that's where I'm going to stop for Chapter 45, and we're going to switch over and talk about Chapter 48, which the focus there is on hematology. So hematology is the study of blood and the tissues that produce it. Blood analysis is one of the most common diagnostic tests performed in a doctor's office. And as a medical assistant, you have to have an understanding of exactly how to correctly collect, handle, package, and analyze blood specimens. But you have to also know what an abnormal result would be so you can inform your provider. So knowing that information, again, what would really be your role with hematology? So again, you're collecting those samples, but please note that performing lab tests is really within your scope of practice. If you've been in your clinical courses, we've been drawing blood and giving it, procedural things done. That's part of your job. You are able to process the sample if there isn't a lab that you send all of your samples to. And you're also responsible for relaying results to your provider, especially those that are abnormal. So now let's discuss blood functions, the formation, and components. The main function of your blood is to transport and protect. The blood takes oxygen and nutrients from the body and removes carbon dioxide. It carries your white blood cells to fight off infections and platelets to help you during a healing process. But blood also helps you to regulate your body temperature. And blood is composed of plasma and formed elements. Plasma is really the liquid component of your blood, and it makes up about 55% of your blood. 90% of plasma is really water, and the other 10% is solid substances. And the formed elements in your blood are things such as red blood cells, white blood cells, and platelets. Red blood cells are also called erythrocytes, white blood cells are called leukocytes, and platelets are called thrombocytes. And there are some common tests that you'll deal with. These are things that you've probably been talking about all along and we've talked about before. And your homework assignment this week. One written assignment about bacteria and their shapes, due August 27th.